Hey, Hammy here, back with another section, another video over our chapter over cell communication. How do cells talk and communicate with each other? This is actually part 4 of this video series over cell communication. In this video, we want to look at the transduction. Remember that we break cell communication into the 3 main parts, okay, where you have the signal reception, and then the transduction through the cell, and then you get the cellular response. So part 4 here is talking about after the signal is received, the transduction of that signal. How is that signal relayed through the cell to create a cellular response? Okay, signal transduction. It's important to understand that it's not just, okay, here is the reception, okay, of our signal or our ligand. Okay, and then it's transduction that goes through the cell, and then we get a cellular response. That oftentimes this isn't just one sort of protein that's shuttling the response. Okay, this is often multiple steps, multiple proteins that affect each other, and you'll see that here in a second. These multiple pathways can amplify, okay, so they can spread out when one can spread out to another, to another, to another, and it can amplify that response, okay, and make that response much larger than just a single protein can. Multistep pathways provide more opportunities for coordination and regulation, for the cell to coordinate its response in a more timely manner, and also getting different parts of the DNA to create a cellular response, or to create that certain response that is needed by the cell. The molecules that relay or transduce this signal from receptor to response are most likely proteins. And like falling dominoes that are kind of set up, and when one falls, that causes the next, the next, the next, the next. Okay, one activates another. Okay, and on and on and on until you get to the response step. At each step, the signal is transduced into a different form. Okay, and most of the time we're talking about a shape change in the protein. In a lot of pathways, this shape change, this signal, is a cascade of phosphorylations. Okay, phosphorylations, adding phosphate groups to things. Okay, the class of proteins that does this is called, or enzymes are called protein kinases. Okay, protein kinases. Kinases are enzymes that take phosphates off of ATP and attach it to a protein. Okay, this process is called phosphorylation. Okay, so these kinases are going to phosphorylate, grab the phosphate off an ATP, and then attach it to a protein somewhere else. The opposite, or dephosphorylation, is done by protein phosphatases. Okay, enzymes that remove phosphates are phosphatases. Okay, so we're going to have kinases, which are going to add, and phosphatases that are going to remove phosphate groups. Okay, this phosphorylation, dephosphorylation acts kind of like you would think of a light switch, and that light switch, you can flip it on, you can flip it off. Okay, so if you're going to add phosphate or take phosphate off, it's going to activate or deactivate that particular protein that's part of the transduction pathway. Okay, this image is kind of showing an overview of that. We're up here. Here's our reception up here from our ligand. Okay, and when that happens, and this can be GPCRs or it can be TRKs, those sort of things. Those both can be stimulated by a ligand to activate a kinase. Okay, so here's inactive kinase. Okay, they're going to use to activate the kinase. Okay, that active protein kinase is going. Here's our ATP. It's going to remove a phosphate group, so now you just have ADP, and that's going to take protein kinase 2. And if you look over here, here's our phosphate group. Once you add the phosphate group, it becomes active. Now, once that is active, it's going to take another ATP, and it's going to add that ATP to the next step, or the third step in our transduction pathway. And on we go until we activate some sort of protein, or it might be a transcription factor or something like that that can trigger a cellular response. And we'll get into that cellular responses in part 5 in the next video. Oftentimes, as part of the transduction pathway, we've got these second messengers. Okay, second messengers, the first messenger is usually the ligand that's attaching to the receptor in the cell membrane. Second messengers are small, non-protein, water-soluble molecules or ions that are usually in the cytoplasm. Okay, that can spread throughout the cytoplasm by diffusion, and it can participate in these pathways that were initially initiated by the GPCRs and the RTKs. Two common ones we want to look at today are cyclic AMP and also calcium ions, Ca2+. These are very common second messengers. Okay, the first second messenger we want to kind of look at is cyclic AMP. Okay, cyclic AMP is one of the most widely used ones as a second messenger. And when an enzyme in the plasma membrane gets triggered by reception by a ligand and a RTK or GCPR might take the G protein that shuttles across will then activate the enzyme adenylyl cyclase. Okay, adenylyl cyclase. Adenylyl cyclase will convert ATP, adenosine triphosphate, into cyclic AMP. Okay, in the next slide here, we'll look more specifically at how this is different than ATP. Okay, if you take a look over here on the left, okay, this is our ATP, okay, where we have our adenine, and down here is our ribose sugar. So that would be these two would be our adenosine, and then we have our triphosphate. What adenylyl cyclase does is it removes, these two phosphates, takes this third phosphate here and connects it back to the carbon on the ribose. So it's cyclic because it reconnects back to the ribose over here. So cyclic AMP, adenosine monophosphate, and it's cyclic because that phosphate is attached to the ribose in two places and kind of makes a circle. Okay, so adenylyl cyclase will remove those two phosphates off of ATP and make cyclic AMP. The opposite arrow, that would just change it a little bit differently with the addition of water, phosphodiesterase will break that bond and turn it into just regular adenosine monophosphate. So you'll see this adenylyl cyclase and also phosphodiesterase when we look at this pathway here in a second. There are many signal molecules that can trigger the formation of cyclic AMP. Other components of the cyclic AMP pathways are G proteins or GPCRs and protein kinases. Usually cyclic AMP will activate protein kinase A, which then, remember we said we talked about them kind of being like domino setup. That phosphorylates other various proteins, and then it's kind of part of that transduction pathway that dominoes throughout the cell. Further regulation of cell metabolism is provided by G protein systems that will go back and inhibit the adenylyl cyclase so that it will stop the cyclic AMP from working. Okay, so this is just kind of a brief overview. So here's our GPCR, our G protein coupled receptor. Okay, here comes a ligand such as epinephrine. Okay, that's going to allow GTP to activate the G protein. The G protein is going to slide across the membrane and activate adenylyl cyclase. Adenylyl cyclase is going to take ATP, change it to cyclic AMP. Okay, so now cyclic AMP is considered our second messenger. First messenger is out here, our ligand like epinephrine. And our second messenger, cyclic AMP, then is going to cause this cascade: protein kinase A, kinase B, kinase C, kinase D, all the way till we get our response. The second messenger that are really, really common in cells are calcium ions. Okay, the calcium ions is important because that has to do more with concentration of the calcium ion. If we kind of look at a typical cell, here's extracellular fluid, nucleus, cytosol or cytoplasm, and mitochondria, the endoplasmic reticulum here. You can see down here at the key, high calcium. Usually you read that the calcium ion concentration outside the cell can be 10,000 times what you find inside the cell. Okay, why is that? Well, there are calcium pumps that pump calcium ions out of the cell. There are calcium pump ions that pump them into the matrix of the mitochondria or into the lumen or the empty space in the endoplasmic reticulum. Okay, so the calcium levels in the cytosol tend to be very low, whereas in the ER, in the mitochondrial matrix and the extracellular fluid, calcium levels are very, very high. What we see happen is when a ligand or signal relay by signal transduction pathway triggers this calcium release in the cytosol, we see pathways that involve inositol triphosphate and disacylglycerol. Okay, disacylglycerol. Or I will often just refer to them because those are really hard names to say. It's IP3 and DAG. So I call them IP3 and DAG will act as our second messengers. Okay? And that's often comes from an increase in calcium or causes an increase in calcium, which then will cause a cellular response. Okay, here we can kind of see that happening. Okay, here comes our ligand such as epinephrine. Okay, attaching to a GPCR. Okay, the G protein is going to be, a GTP is going to activate the G protein, okay, which is going to activate phospholipase C. Phospholipase, okay, these are little phospholipids. Remember, this is a phospholipid bilayer, so you've got little phospholipids here. Okay, there's a phospholipid called PIP2. The phospholipase C is going to cleave this bond here from DAG and IP3. Okay, so it's going to split PIP2 into DAG and IP3. This IP3 is going to act as a second messenger, travel throughout the cytoplasm to little channels, IP3 gated calcium channels. When IP3 binds to this channel, okay, it will open up, and calcium ions, which are in a high concentration in the ER lumen, will rush out into the cytoplasm. Okay, this change in concentration is then going to activate other proteins and then cause our cellular responses. Okay, in our next video, okay, in part 5, we want to look at what are these cellular responses. What are some of these typical cellular responses that happen at the end of a signal transduction pathway?